morning, everyone. My name is Lea Sistonen, and I have this privilege to welcome you all to the 8th International CSSI Congress on stress management mechanisms and pathways. So this is the 8th in a row this society has organized worldwide, and we are really, really happy and proud of having you all here in Turku. It's amazing actually that you all are here. I know that you have been traveling, some of you like many days and nights. Some of you just come over the river or cross the street. So it's a great mix. And I think that is also describing the meeting. We really would like to have a good mix of speakers, attendees, all kinds of presentations, and also the social program. On the behalf of this society, I would just want to emphasize a couple of things, that it has started as a demand to put together a society which is really like tolerating all kinds of topics, all related to stress biology and biomedicine. And we know that there are many, many meetings around the world on different subtopics of stress biology and biomedicine. But I think this society really nurtures the versatility, diversity, and also try to be on the pulse where these kinds of research topics are heading also in the future. And that's why in this program we have a mixture of really young scientists. This is maybe the first international congress for some of you. I know there are some students that this is absolutely the first time they are attending an international congress. And then there are also participants who have attended quite a few congresses before coming to this one. So maybe that is also something which is telling about how this society nurtures the science also for the future. So the subtitle of the meeting is Stress Management Mechanisms and Pathways. It's a very wide title, and as you have seen, if you have opened the book, the program book, the topics are all about how to manage stress in nature and also in our biological systems, and how they are disrupted in different diseases, and how these topics actually could be used also for the benefit of the mankind for different purposes. So that's why we really try to highlight the breakthroughs that have been made and to be made in the near future, and also discuss them here commonly. So we don't only have these talks, the plenary talks here, we have the poster sessions, and also we try to have long enough breaks that there will be possibilities to discuss with your old friends and colleagues, and also make new friends and maybe become also colleagues in the future. So Helen will be remembered also after the meeting. So, I said, there's a committee, but then there are all these other people who we need. So you have already seen quite a few of these young people at the front desk. They will help here with the technical part of the meeting, and they are really eager to be part of this organization. So I just highlight the group of John Eriksson in this winter-like picture, and then my own group, more like in a green background. So many of these young people and also others are here to help. They have those t-shirts of our university, but there are also other people. And please don't hesitate to ask. So we are here for you, you just have to ask. And the final thing I want to emphasize that, again, of course, there's a lot of manpower behind it, but we also need some money. And without financial support, again, we would not be here. So I would like to thank everybody who has contributed. So the society CSSI is the main sponsor, of course, of this meeting. Our university, Åbo Academy University, and particularly its foundation. So Stiftelsen för Åbo Academy, as it's called in Swedish. So it's a Swedish-speaking university in Finland where we work. So they contributed, and for instance, the whole social program is supported by our university. And then, of course, we have Springer Nature, Stressmark, MGM Resorts, very generously supporting our activities. And then the local forces, Turku Bioimaging and Euro Bioimaging. If you want to know anything more about these organizations, so John Eriksson here, could you please stand up? He's very happy to tell you about that. Those organizations, Biocity Turku is of course very instrumental, and finally Science Park of Turku has also supported our meeting. So we are very grateful and hope that you all enjoy what we have been able to get from these supports. And with that, I just wish you wonderful Congress, enjoy every moment, every aspect of it, and please come to us and tell how we could improve it as long as we go along. So happy Congress to everyone.
Good morning and welcome to all. I'm Gabriella Santoro, and I have the honor today to be as the president of the Cell Stress Society to award the Cell Stress Society Medallion to Larry Hightower. So this is the medallion. You see the symbol of stress. If you are wondering what's in the center, that's a mitochondria. Larry is going to tell us the mitochondrion story later. I want to start by saying that without Larry, we wouldn't be here today enjoying a beautiful Turku, and what it looks like is going to be a very stimulating, very rich program. And by the way, Lea, thank you for hosting us and organizing such a nice meeting. So a few words about the medallion. The first medallion was given in the year 2000 to Takashi Yura, and the second to Susan Lindquist in 2003. I know we have a whole session with Susan, but I just want to say a few words because we were all so shocked when we heard the news that she's not with us anymore. Many of us were at the meeting in Old Town Alexandria last October, and we heard about Susan. So I just want to say that definitely Susan has been one of the pioneers in the field of the HO proteins, first working mainly in yeast, but then lately also in human cells, and she made a really major contribution, among other things, in understanding some of the crosstalk between the heat shock response and metabolic stress, which I think is very important. But let's go on. Then we have in 2005 Aaron Chikanover, and in 2007 John Ellis, 2009 Costa Gorgopoulos, and in 2015 Kazutoshi Mori, and last but not least my good friend Ferruccio Ritosa, who got the award in 2010. And most of you, probably all of you, know that Ferruccio is considered the father of the heat shock response fields that he discovered more than 50 years ago in Italy. Ferruccio is quite an exceptional guy. He's a very acute mind, very independent person, I would say very courageous, and really kind of a symbolic scientist. At some point, he decided to leave science for becoming a sculptor for art. And I like to think about Ferruccio as the man who transformed a laboratory accident, because maybe not all of you know that this whole field was born by a malfunctioning incubator in Ferruccio's Drosophila laboratory. and one of the most prominent, I would say, inexhaustible fields of science that we work in. So I'm mentioning Ferruccio because it was because of Ferruccio that I started to know Larry Hightower much better. I knew Larry as a scientist, I knew your work, and I knew him as the president and the founder of the CSSI Society. He's a great organizer, but then during this, we organized a meeting together, a meeting that was very much wanted by Larry to honor Ferruccio and to celebrate the 50th anniversary of the discovery of the heat shock response. And we had the way to interact more, so I started to know Larry better as a person. And I will not forget that here, and we had this meeting in Rome at the Arapacis, and under the smile of the goddess of peace, Larry gave one of the most beautiful speech, reminding us as art and science are the forces that drive people together, that connect people from different countries independently of the political situation, and it doesn't matter how difficult it is, we are on the same wavelength. And during really this meeting, I learned that Larry is not only a great scientist, a great organizer, but is a great personality. He's a great passion for science, an extremely open and positive attitude. I don't think I've ever seen you upset, Larry. I don't know if any of you have seen Larry upset, maybe Gail, I haven't realized. So this is Ferruccio and Larry, and this was when he was speaking. And I'm not going to talk too much about the career achievements of Larry because we have a surprise for Larry. Antonio De Maio is going to tell us the real story. I just want to mention that after his degree in chemistry, Larry got his PhD in biochemistry at the University of Harvard. And after 1 year in the Department of Microbiology, where he worked with viruses, actually he was one of the first scientists that studied the heat shock response during viral infection. I think it was paramyxoviruses mainly, yeah. Then he became first assistant professor and then professor and head of the Department of Molecular and Cellular Biology at the University of Connecticut, where he still is as a professor emeritus. And during his career, he got many honors. These are a few. I want to point out this one from the American Association for the Advancement of Science, because it was given to him, I didn't know about this, Larry, but for his work on proteotoxic stress, but mainly for the work on the heat shock response in the desert fish. I didn't even know there was a desert fish, so maybe you can tell us about that. But Larry is very, very interested in the environment, and in fact now he's the biology advisor for the NGM International Program on the effect on environmental stress on endangered species. So this is another aspect of Larry. And so we can add now to this list the CSSI Society Medallion in recognition of these discoveries about heat shock proteins, thermotolerance, and proteotoxic stress response. And this is the certificate, but not before we hear the real story by Antonio. Oh, Antonio.
Thank you, Gabriella. I have an honor that Gabriella asked me to talk about Larry because during the last year we have interacted a lot. So I think I know Larry very well. Now let me start with a little story. When Larry traveled to these meetings, Gail, Larry's wife, sent me the itinerary of the trip. And I have to arrange my trip. So I arrived before them. So I set the ground so when they arrive, they don't get lost. And I've been doing that for a while, and I now realize that I am Larry's and Gail travel chaperone. So, can we have the lights off, please? So, Larry, the multifaceted man, the real story. This is Larry the baby. And this is Larry the child. Now look at that, he is fishing there. So I think this moment set her career as a scientist at this early age. And also here we have Larry the young boy. But this is very surprising to you. Larry the athlete. And this is during the high school. He was part of the field and track team at his local high school. This is Larry the PhD student. You see, he knows how to type it. And this is Larry the warrior. And notice that he has a gun in one hand and a set of books in the other one. You know, this is great. This is Larry the doctor. And Larry the stylish professor. And look at that tie. That tie tells us about his research because in fact, he publishes papers on the stress response in fish. This is also Larry the friend, Larry the zookeeper, and you see this has also changed what he's doing now. How he's working with NG Entertainment because, you know, he's been involved in, he had this zookeeper job before. But more important than all is Larry the pioneer. Because Larry published the first two papers over 30 years ago. He was the first to say that Hitchcock proteins could be released from cells and bind lipids. And nobody paid attention to what he was doing after many years after, but he is the first one to show that. So that's what I always call him, for the discovery of the extracellular HSP70 and his capacity to bind lipids, the Christopher Columbus of extracellular Hitchcock proteins. Because he discovered them, but he doesn't know what he was doing. With that, please, Larry Hightower, recipient of the CSSI International Medallion for Career Achievement.
Here is the medallion, certificate later.
So before you start, maybe you take a picture of the three of you.
Or you can pose a little bit.
Sorry.
Looking good.
Come on, John.
Thanks.
Well, thank you so much. I really want to start by thanking Gabriella and the council for bestowing this honor on me. It's both exciting and humbling. It's humbling because the people I've now joined, you've seen up here, the previous winners, they're all fantastic scientists and great friends, and I learned much from each of them over the years. So that makes it feel very special. When I heard that Antonio was going to tell my life story, I went ahead and quickly wrote out my entire speech and put it in the abstract book. I knew there wouldn't be much time left to talk, but that's okay because then I could reason things out, and I thanked a lot of people in there. So I decided what I wanted to do here today is just speak from the mind and the heart, and just tell say 4 very short stories that hopefully will give a flavor for my 40-year career, which I can't believe 40 years ago I did my first experiment that got me into this field. But let me just say one thing about one of the slides Antonio showed. Yes, I was drafted into the Army after my first semester at Harvard, and it emerged that there was only one way I was going to get back to Harvard, and that path led through Vietnam. So, like almost all the students I knew at the time and the faculty, I was very much against the war. It was a terrible war. It should have never been fought. It was just a horrendous waste of life and resources. But it happened, and but I will tell you that that was a Army-issued M14 rifle, and I'm proud to say that I never shot it at anyone during my 13-month tour there. In fact, I was also issued an IBM Selectric typewriter because the Army was in more need of people who could write and spell than they actually were people who could shoot that big rifle. The book was not a manual on how to use the rifle. The books I held were from my department secretary, Thelma Shapiro, who was kind enough to send those to me so I could study for when I got back to Harvard. And the other thing was that, so I was the company commander's clerk, and I was the sergeant major's jeep driver. And so if you know the old series MASH, which is one of my favorite series with Alvin Holda, the Korean War, well, I was Radar O'Reilly. So if you remember Radar, that was me. But because I was Radar O'Reilly, a time came in 1970 when I needed to get to Saigon. And it was not easy to travel at that time, and so I had to get permission and leave from the company commander, and since he was my boss, it was easy to do. And the reason I needed to go to Saigon was I needed to meet with Professor Matthew Messelson, who was one of my teachers at Harvard. But it had really nothing to do with Harvard or science, academic science, really. Messelson was there as part of a USAID project studying the use of Agent Orange. And he had this hypothesis that the US military was not just spraying enemy crops, which is what they claimed they were doing, but that they were actually spraying civilian rice fields as well. And he used the Army's own maps to prove that. So I met with Matt in 1970 during the war at the old Hotel Continental in the center of Saigon. And we discussed how, I might help him obtain information that might be helpful in his study. And he told me that he was going out into the field quite regularly on military helicopters with Marine bodyguards, looking around in the south of Vietnam for evidence of damage. And he wanted to get into the mangrove swamps because he had the idea that even though the Army said the mangrove trees had been sprayed and they would come back, he had the idea that scrub bamboo was growing much faster and that was going to choke out the mangroves, and he was right there as well. But he also complained to me that he wasn't happy with the helicopter crew because the Marines wouldn't land in the mangrove swamps where he wanted them to land. And I had to gently tell Matt that you don't want to land and get out where the Marines still want to go. And as soldiers, we have a rule, As Army soldiers, we had a rule: if the Marines won't go there, you don't go there. So, but anyway, when I got back to Cambridge, Matt was all going around town telling this great story about the use of Agent Orange and the misuse of Agent Orange. And the reason I tell you this is that I credit Matt with really showing me by example what you could do with your academic skills and your knowledge on the social stage and the political stage. And professional service has been a big part of my career during my entire, however, 40 years or so. And I really credit Matt with showing what could be done. So, in terms of the science, I think one of the amazing things is that 40 years ago, there were two observations made. I made one of them. And a man named Fred White, who was a professor at Memorial University of Newfoundland, made the other one. The one I made was really quite simple, but the human interactions that it contained were not. And when I thought back on it and said, I wonder what's still applicable today, because so much has changed, how we work in the lab and how we plan experiments and what we do. So what maybe hasn't changed? Well, maybe what hasn't changed are some of the human interactions. And it came to mind that famous statement by Louis Pasteur, chance favors only the prepared mind, which I understand from my French friends is the better translation. But I also learned that the observation, the experiment was very simple. It was a virology experiment. I was using amino acid analogs to try to block the processing of precursor viral proteins, and we knew there was at least one fusion glycoprotein was made as a processed precursor, and I wanted to look for other ones. And so I was treating infected chicken embryo cells with the amino acid analog cannabinine, which is an arginine analog, and we suspected that it was either a trypsin or a chymotrypsin-like activity, so that would be a good one to use to block the processing sites. What?
Microphone.
Oh yeah, and the thing was that in the uninfected analog-treated control, the whole pattern of protein synthesis changed on gels. We were running SDS page gels to see it. And it was amazing. It was an unbelievable change. It was just 4 peaks had taken over the entire gel, and the main one was at 70K. And I had only seen that change that fast in a eukaryotic cell in terminal differentiation situations, like the differentiation of the particular site, the red blood cell, or in very lytic viral infections. NDV infection, for example, shut down post-protein synthesis and only a few polypeptides are made. So I knew it could happen, but couldn't imagine how it was going on there. Now, I knew how to interpret the analog data because my mind had been prepared by associating with a young professor at Harvard Medical School who was in the physiology and cell biology department, which was right next door to my lab, which was in the micro department, Michael Bratt's lab, which was a virology lab. And this young professor was Alfred Goldberg. And Fred got interested in my project because it was protein metabolism. And he invited me to his weekly sessions, which were combinations of lunches and lab meetings. And that's where I learned how to use and interpret analogs, because he was doing a lot of that, and he was one of the few professors in Boston that was actually seriously interested in protein degradation, or protein turnover as we called it, because he was studying diseases of muscle wasting at the time, and that's a protein degradation issue. And he was the only one I knew who firmly believed that protein degradation was regulated. Everybody else thought it was taking out the molecular trash and too boring to be studied. But he was convinced it wasn't. So we kept up. We met every once in a while. Fred came to University of Connecticut every 2 years to participate in a very well-known seminar series, Biological Regulatory Mechanisms, by one of my colleagues, Ed Carolla there. And he immediately went for the idea and did a tremendous job spreading the idea. And the particularly radical piece of it was when I proposed that cells could sense the presence of abnormal proteins and distinguish between the normal and the abnormal state. And most everyone I talked to about that thought it was crazy talk. And but Fred didn't, and he went on to really, along with his students, do some very nice experiments to help demonstrate that cells really could sense and distinguish normal and abnormal proteins. Then the other observation was made by Fred White up in Memorial University in Newfoundland. And Fred White was a Henry Mahler student. Back in my days, if you were studying biochemistry, you studied Mahler and Cordes. And what we used to say as students is if you could actually survive reading Mahler and Cordes and still were interested in biochemistry, you were going to be a good biochemist. And so Fred was slicing brains. He was a brain biochemist, and he was slicing rat brains in slices and incubating them in vitro and pulsing in radioactive amino acids, and thinking he was going to study normal brain protein synthesis in a more accessible format. But he had a problem. And my colleague at UConn had just come from Memorial University, Barry Hall. And he knew Fred very well. They were close family friends, and they'd gotten into this habit of meeting in the summers in Vermont for a little family vacation. And as Barry tells me the story, and of course Barry knew all my stuff because Barry loved to think about science, and nothing could be too outrageous or unusual for him not to want to think about it. So this was just fine with him. So they were up in the cabin one summer evening doing their little family vacation thing. And as Barry tells it, there was a very large flask of Gallo Hardy Burgundy wine involved in this. And it was late in the evening, and a lot of wine had been imbibed. And Fred started, pouring his heart out about these horrible proteins. That there were about 4 of them, and he just couldn't keep them off and suppressed, and they were ruining the normal pattern of brain proteins. And he was pretty upset about it. And so Barry's response was, "Well, Fred, how big are they?" And Fred said, "Well, the most prominent one is about 70 kilodaltons." To which Barry said, "Well, Fred, that sounds like Larry Hightower's proteins." Now, he didn't say that, sounded like the heat shock proteins because he didn't know about the heat shock proteins. And I couldn't tell him about them because I didn't know about them. I'd never heard of the heat shock proteins. And it was 2 years I studied what I thought were analog-induced proteins, but we very quickly found that they only worked as an inducer if they were incorporated into the protein, and a few simple experiments, good control experiments, showed that it was really the analog substituted abnormal misfolded proteins that were being sensed. But anyway, Fred came to visit Barry a couple weeks after that. And Barry brought him over to see me, and Fred thought it was going to be quite a short meeting. But we talked for 2 hours and we traded samples, and the bottom line is, of course, that they were the same proteins. We did tryptic mapping and all together and ran them out on the same gels side by side and mapped them, and they were the same proteins. And so that really got us started and got us thinking about physiological functions for these proteins. And Fred was quite certain that it was trauma that was inducing them. It was the trauma of slicing or the initial inflammation. But he manipulated the oxidative Krebs ringer quite a lot and couldn't change the fact that they came on. So we suspected it was the trauma. Basically, it was a wound response. And his postdoc, Bill Curry, was put to the task of finding out where in rats these things were made. And he's going to tell us about that tomorrow and show you some pictures of himself and Fred in those days. But the bottom line is many organs, virtually all organs, have these proteins in a heat-shock rat. And mostly they congregated in microvasculature. So we knew in the late 70s that if you wanted to study these proteins physiologically in animals, you had to be looking at the microvessels and the endothelium, the vascular endothelium. And that's still true today, and that's still where I'm looking with my clinical colleagues for these proteins and how they're functioning. So the amazing thing is, 40 years later today, we're still basing our experiments on those observations. Now, the most recent thing in my last story has to do with Susan Lindquist. And she introduced me to a young MD, PhD, who'd received his PhD in cellular immunology at Chicago, and that's where he met Susan. And he did this remarkable experiment with rat kidneys where he chocked them before he harvested them and put them on the shelf to mimic transplantation, holding storage and transplantation. He was able to double the shelf life of the rat kidney from 24 to 48 hours by this heat shocking. And he had heard a lecture or two from Susan and thought they might be the heat shock proteins, and he carted some samples over to her lab and she assayed them for him. And when he decided to come back to Hartford, because he had trained at UConn Health, Susan said, "You have to meet Larry Hightower. I'll make introductions. I'll let him know you're coming, what you're doing." And that's how we got together. And we've been working for the past 20, 25 years on how to stress condition humans, how to stress condition patients. And we know how to do that now. We have to prove what we're doing to the point where other physicians will use it. But there are people walking around right now who've been stress conditioned, and their physicians don't know they did it, and the patients don't know they have. And that's perfect for our field, right? That makes perfect sense. But the stress conditioning is done by hyperbaric oxygen therapy, putting patients in a chamber at 2.4 atmospheres, and basically inducing a little, we don't call it oxidative stress, we call it oxidative signaling. You know why. And it just turns on NRF2 like there was no tomorrow. And it turns on a little bit of SF1, so you get just enough HSD70 marker to tell you, yep, that piece of it, the proteotoxic stress response network is coming on a little bit. But mostly it's the NRF2 guys, prominent among them being metallothionein and hemoxygen, ACE1. And we kept talking now about cellular energy homeostasis. And we decided, well, we have to do better than that because we've got this beautiful term now, proteostasis and the proteostasis networks. And now we call the other side of it caloristasis, the caloristasis network, because calorie is such a beautiful, substitute for heat and the way biologists and chemists like to measure heat anyway. And the physicists are stuck with their ergs and joules, but that's okay. They can deal with it. So we're having a great time with that. And George is now occupying an endowed chair at UCSD. It's the first endowed chair of hyperbaric medicine we know of in the U.S. And he's organizing a clinical trial, hard at work with that, which is why he hasn't met at this meeting. And but we're already pretty convinced. We have markers, urinary markers now, for some of the oxidative pathways that should be turned on by NRF2, and so we can follow the path now in humans. Because these techniques for hyperbaric oxygen therapy, they're FDA approved, but they were put together to treat mainly chronic diabetic leg ulcers. And the thing that is very clear that they do is stimulate angiogenesis. So we're back to microvessels. We're back to microvessels again. And now we're also with caloristasis and proteostasis. So I'm having a grand time, and it's going to be a great way to go out. And I'm excited for our field. And I'm very happy that you all are here and are interested in our field, because I truly believe the best days and the most exciting days are ahead of us.